From the simplest bacteria to the largest whale, every living thing on Earth is made of cells, the basic unit of life. And all of those cells contain instructions written in the language of DNA. But how does a single strand of DNA control an entire body? That's what we're going to find out in this episode. Welcome to this amazing channel. All living things are made of cells, but not all cells are alike. Muscle cells enable movement. Nerve cells carry messages. Some cells have a single job and others have many. But every type of cell needs a full set of genetic instructions. And that's where DNA comes in. DNA is a molecule that carries genetic instructions in all living things. Think of it like a language. Just like English has 26 letters that combine to form words. DNA has 4 letters, A, T, C, and G. These 4 letters form words called genes. Every possible combination of these 4 letters spells out a unique set of instructions. So, just like there are an infinite number of words you can make with 26 letters. The same is true with DNA's 4 letters. There are countless possible combinations. In fact, the entire collection of genes in your body contains over 3 billion letters. These genes are organized into structures called chromosomes. Humans normally have 46 individual chromosomes packed inside the nucleus of every cell. However, before a cell divides, it must first duplicate its entire set of chromosomes. This ensures that each new cell will have a complete set of genetic instructions. And this process of copying DNA is called replication. Let's take a closer look at how it works. Here's a model of a chromosome with its 2 identical sister chromatides joined together at the center. The enzyme helicase comes in and unwinds the double helix, separating the 2 strands. Each strand then serves as a template for creating a new complementary strand. Free nucleotides in the nucleus pair up with their complementary bases on each template strand. On one strand, adenine always pairs with thymine, and cytosine always pairs with guanine. Once the new strands are formed, the enzyme ligase comes in to join the fragments together. And the result is 2 identical copies of the original chromosome, each consisting of 1 old strand and 1 new strand. Now that the genetic material has been duplicated, the cell can divide and create 2 identical daughter cells. But remember, genes are the instructions. They're like the words in a book. They don't do anything on their own. It's the proteins that actually run the cell. But how do genes get from the nucleus to the rest of the cell? To find out, let's start with this gene here, which codes for a protein that determines eye color. First, the gene unwinds and separates the 2 strands of DNA. Then an enzyme called RNA polymerase binds to the gene and starts reading the sequence of letters. As it reads, it creates a complementary strand of RNA. Remember, RNA is a similar molecule to DNA, except it uses the sugar ribose instead of deoxyribose, and it uses the base uracil instead of thymine. Anyway, the RNA polymerase reads the DNA sequence and builds a matching RNA strand. This process is called transcription. Once the RNA polymerase reaches the end of the gene, the newly formed RNA strand is released. This RNA strand is called messenger RNA, or mRNA for short. The mRNA then leaves the nucleus and enters the cytoplasm where ribosomes are waiting. Ribosomes bind to the mRNA and begin reading the sequence of letters. Remember, DNA uses the 4-letter code of a T, C, and G, but the language of proteins is a 3-letter code. That's because groups of 3 letters in mRNA, called codons, code for specific amino acids. Each codon specifies which amino acid should be added to the growing chain. The process of making a protein from mRNA instructions is called translation. Here's a chart that shows all the possible codons and their corresponding amino acids. You can see that the codon GCA codes for the amino acid alanine. If the ribosome encounters the codon GCU, it will add the amino acid serine to the protein chain. It continues reading the mRNA and adding amino acids until it reaches a stop codon. at which point it releases the completed protein. The newly made protein will fold into a unique 3D shape that enables it to carry out a specific function. So that's how a gene works. It goes from being a sequence of letters to a functional protein that carries out a specific task. Genes come in two different flavors, those that code for proteins and those that don't. Those that don't usually regulate the activity of other genes. Now let's go back to our original question. How does a single strand of DNA control an entire body? Well, here's a simple analogy. Think of the DNA as a library, genes as individual books, and traits as the stories contained within those books. For example, eye color, height, and blood type are all traits. But what determines whether or not you have blue eyes, are tall, or have type A blood? Are the genes themselves. Each trait is controlled by multiple genes. In fact, most traits aren't encoded in a single gene. Instead, they're polygenic, meaning they're controlled by many. Take eye color, for example. Two genes work together to determine your eye color. One gene controls the amount of melanin produced, while the other controls how that melanin is distributed. That's why you can have two different eye colors, one for the melanin concentration and one for the melanin distribution. You inherit two versions of each gene, one from each parent. So you could inherit brown eyes from both parents but still end up with blue eyes. That's because you only need one dominant allele to express that trait. In this case, brown eyes are dominant over blue. And now you know how a single strand of DNA controls your entire body. Every trait you have is controlled by a unique set of genes. And every gene is a segment of DNA. So ultimately, everything about you is controlled by your DNA. Thanks for watching.